Hello everyone, welcome to Get My Class. Today we are going to learn a topic from a chapter that is molecular basis of inheritance. One of the most important chapter with respect to your board exam as well as your any kind of entrance exam, whether it is NEET or any other medical entrance exam. So let us see the topic for today. What we are going to learn from the chapter that is molecular basis of inheritance is nucleic acids. So the topic for today is what? Nucleic acid, under which we are going to learn about DNA and RNA. So let's begin. So what are nucleic acids? Nucleic acids, as the name indicates, they are the type of acids which are present within the nucleus. Okay, so that's why they are called as nucleic acid. Okay, but then they are present within the nucleus in case of eukaryotes. But in case of prokaryotes, they don't have nucleus. They have something called as nucleoids, which we are going to see. So when we talk about nucleic acid, there are two types of nucleic acid which are found in living organisms. One is DNA, the other one is RNA. So DNA is nothing but the deoxyribonucleic acid. RNA is nothing but ribonucleic acid. Now, what is DNA? As I told you, that is deoxyribonucleic acid. What is it? Deoxyribonucleic acid. Nucleic acid. Since the name says it is deoxyribonucleic acid, it is made up of deoxyribonucleotides. So it is a long polymer. Polymer means it consists of many units of deoxyribonucleic acid. That's what is DNA. Now, the length of the DNA is usually defined as the number of nucleotides present. So, since we know that DNA is nothing but the chain of nucleotides, hence the length of the DNA will be decided by the number of nucleotides present. Okay, for example, in E. coli cells, there are about 4.6 million base pairs. And also in case of human DNA, like in case of human cells, the haploid content of human DNA is about 3.3 million base pairs. Okay. Then, what is so important about DNA? It acts as the genetic material in most of the organisms. Okay, and what is the reason? Because it is more stable, or you can say it is more stable than RNA. Okay, so we will also learn about what is the reason that it is more stable than RNA. Now, the RNA. So I told you that there are two types of nucleic acid. One is DNA and the other one is RNA. The RNA, it is, of course, this is also a long polymer of ribonucleotides. So here, DNA is made up of deoxyribonucleotides, whereas RNA is made up of ribonucleotides. So here the speciality is, or the difference is, it has additional OH group at the 2' carbon, okay, within its sugar, that is the ribose sugar. So we are going to learn about the structure where you will come to know about the presence of OH group at 2' position in RNA, whereas in case of DNA at the same position, a hydroxyl group is not there, instead only hydrogen is there. So there is removal of oxygen in case of DNA. Now, RNA also acts as genetic material in some of the viruses, okay? And of course, they act as messengers. So they carry the information which is there in the DNA, okay? And they transcribe it, it forms RNA, and further RNA will be utilized for the formation of the protein. So that is why we can say that it is the messenger in the sense it carries the information from the DNA, and it will be helpful in the formation of the protein. Then it functions as an adapter. Okay. Then so you have different types of RNA, like the tRNA functions as an adapter, the structural, and in some cases as catalytic molecule. So structural, in case of ribosome, it is a part of the ribosome, that is rRNA is present, rRNA. Okay. And this rRNA, that is ribosomal RNA, also acts as an enzyme. So we call it as ribozyme. What is it? Ribozyme. Okay. So that's about DNA and RNA. Now, when we talk about the structure, I have already told you that it is made up of polynucleotide chain. That is, polynucleotide means many nucleotides are present. Okay, where is it? Yes, many nucleotides are present. Okay. So nucleotides, as I told you, in case of DNA, it is deoxyribonucleotides. Okay, whereas in case of RNA, it is ribonucleotides. Now, when we say that the polymers are, or you could say the nucleotides are combined with each other by the process of polymerization, How are they combined with each other? Because of the formation of various bonds. And what kind of bonds? For example, here you have N-glycosidic linkage, that is the bond by which nitrogenous base is linked to the pentose sugar. Okay, so the combination of nitrogen base and pentose sugar is called as a nucleoside. When a phosphate group is added, it becomes a nucleotide. So this difference you have to remember. Now, as I told you that this bonding makes it very strong, and that's how it forms a polymer because of the various bonds like N-glycosidic linkage, as well as you have phosphodiester linkage or phosphoester linkage, where phosphate group is linked to 5' hydroxy of the or OH of the nucleoside. Okay, so together, that is phosphate group and nucleoside together, they form nucleotide. Now, you also have other 3' to 5' phosphodiester linkage, where two nucleotides are linked to form dinucleotide. So of course, every nucleotide has to bind with another one, right? And in this direction, they bind, that is 3' to 5' phosphodiester linkage. Then more nucleotides join to form a polynucleotide. So if there are only two nucleotides, this is how they form, by combining or you can say by using this type of a linkage, that is 3' to 5' phosphodiester linkage. And further, the more number of nucleotides will be added to form an entire polynucleotide or the DNA or even RNA. Now, hydrogen bond. Hydrogen bonds, they help to form double-stranded polynucleotide. Okay, because you know that in case of DNA, it is double-stranded. And how these double strands are formed? One polynucleotide and other polynucleotide. In between them, the nitrogen base will be towards inside, and they will bind with hydrogen bonds. Okay, so here you can say single chain of nucleotide. So this is why it is called as polynucleotide, where you have this is one nucleotide with the phosphate we are talking about. This is second nucleotide. This is third nucleotide. This is fourth nucleotide. So this is a continuous chain of nucleotide. Now, when you talk about double strand, you can see that this is one strand and this is another strand. And here they are protruding their nitrogen bases towards inside. And the complementary nitrogen bases, they bind to each other by the formation of hydrogen bonds. Okay, so always you can see that adenine binds with thymine. Okay, and of course it is vice versa, and guanine binds with cytosine. Okay, this is the complementary base pairing, you can say. So that is the structure of the nucleotide as a polynucleotide as well as your double-stranded polynucleotide chain. Now, what are the structural differences between RNA and DNA? So I have already told you that DNA, where the sugar present here, okay, present in DNA is 2-deoxy, D-ribose nucleotides. That means you can say that here the sugar which is present here, it is 2-deoxy. That means it is devoid of an oxygen at 2' position in its D-ribose, that is in its ribose sugar. Whereas in case of RNA, it consists of ribose sugar. Okay, an extra OH group is present at 2' region. And the second point, it contains cytosine and thymine as pyrimidines and guanine and adenine as purine bases. Whereas in case of RNA, cytosine and uracil are present. So instead of thymine, uracil is present, and whereas the guanines and adenine are present as purines. Then double-stranded alpha helix structure. So DNA, of course, it is made up of double-stranded alpha helix structure, which is more stable compared to the single-stranded alpha helix structure of RNA. And there's a very large molecular mass. It has very large molecular mass, whereas since the size itself is less or small, you can say it is having less molecular mass. Now, that is our structural difference. So let us talk about functional difference. How are they different in their function? DNA has a unique property of replication, so it can get replicated, but RNA does not get replicated. So very, very important point. RNA does not get replicated. So this is the where we are talking, in the organism where the genetic material is RNA. In those organisms, RNA will not get replicated. Okay. or duplication of the RNA does not occur. Then B, that is the point B, you will say DNA controls the transmission of hereditary effects, whereas RNA controls the synthesis of the protein, because DNA, based on the information on the DNA only, RNA will be formed, and based on the information on the RNA, proteins will be formed. Okay. Then DNA mutates at slow rate because, as I told you, DNA is more stable because of the absence of OH group at the 2' carbon, right? Whereas RNA mutates at faster rate because it is more reactive. Then it is more stable, less reactive, whereas as I've told you already, RNA is unstable and more reactive. So that is the difference between DNA and RNA with respect to their structures and function. Now let us talk about double helical form of DNA. So that is how the DNA is present in our cells, right? So double helical in the sense because it is made up of two strands. Okay, it is one strand here and another strand here. And this, you can say it is coiled like a helix. That's why we call it as double helix. Okay, and you can see that it forms like a ladder-like structure. And these nitrogen bases, they form like once they bind with each other, that is the complementary base pairing occurs, then they become like racks of the or you can say racks of the ladder. Okay. Now, the structure of double helical DNA was actually proposed by James Watson and Francis Crick, and they did X-ray diffraction studies. Okay, and again, further data was produced by Wilkin and Rosalind Franklin, who proposed this model of the DNA. Okay. Then, what are the salient features? You already know there are two polynucleotide chain. Okay, this is one chain and this is second chain, and they are in anti-parallel way. They are arranged anti-parallel. So if this is 3' to, you can say this is 3' to 5', and this is 5' to 3'. That's why they call it as anti-parallel. Okay. Then the base in two strands are paired by hydrogen bond. That we have already seen that the nitrogen bases are paired by what bond? Hydrogen bond. Okay. Then always A will bind with thymine, adenine will bind with thymine, and guanine will bind with cytosine. Okay. Then each turn has approximately 10 base pairs. Approximately. Each turn has 10 base pairs. And the pitch, okay, the distance between the start of the helix and the end of one turn, you can say, this is nothing but 3.4 nanometer. Okay. And the plane of one base pair stacks over the other in the double helix to form or to confer stability. So the stacking of the base pair actually gives that stability to the DNA. So that's the structure of double helical DNA. Now, Francis Crick proposed a central dogma in molecular biology. So, what is central dogma? DNA codes for DNA codes for RNA, and RNA codes for protein. That is the central dogma. But in case of certain viruses, what happens? This RNA will be the genetic material. In that case, RNA will code for DNA. That is called as reverse transcription. But normally, this is the central dogma: DNA to RNA, RNA to protein. Okay, so this, or you can say coding or formation of RNA using DNA as a template, it is called as transcription. Further, mRNA giving rise to a protein, it is called as translation. Okay, these 2 points are very important. Now, when we talk about packaging of the DNA double helix, we know when we unwind it, it will look like, double helix like a staircase. But how is it packed within the nucleus? Okay, how is it present? How is it so coiled and how is it so compact? Because of the packaging. Okay, there is a way it is packed and it is coiled and coiled and coiled so as to fit in one particular nucleus in case of eukaryotes, whereas in case of prokaryotes, though they don't have nucleus, but still the genetic material is set at one place. Okay, though it is in cytoplasm, but still they are present in specific region called as nucleoids. Okay, so nucleoids are present in prokaryotes. So there is not well-defined nucleus. That is why they are called as prokaryote. Pro means primitive. Karyote means carry on. That is nucleus. Okay. And here the negatively charged DNA is combined with some positively charged protein. So here also proteins are involved, which will bind to the genetic material or the DNA and keep it in one place within the cytoplasm. Whereas in eukaryotes, we have very well developed nucleus. And within the nucleus, the DNA is coiled around or associated with histone proteins, which are positively charged proteins. Okay. Why? Because they have rich amount of lysine and arginine, which are the basic proteins. Now, why am I talking about basic amino acid? So basic amino acid, these are okay. Why am I talking about basic amino acid? Because DNA is what? DNA is acidic. That's why we say it is deoxyribonucleic acid. Acidic. So this acidic DNA will be easily binding up with histone proteins because of the presence of the basic amino acid. So that is the polarity, that is the attraction, you can say. Then, when we're talking about association of histone proteins with DNA, we are actually talking about the histone octamer, which is very important, around which the DNA is coiled. Okay, and that structure we call it as the nucleosome. What do we call it as? Nucleosome, where the DNA is surrounded around the octamer of histone protein. So this is an octamer. Octamer means it consists of 8 subunits. Okay, which are the 8 subunits? H2A, H2B, H3, and H4. Okay, all in 2 numbers. Then further, they have one histone 1 protein, which acts as a linker protein, linking the 2 ends of the DNA, which surrounds the octamer. Then, so nucleosome is formed by the combination of the DNA and the entire octamer. Now, okay, single nucleosome contains about 200 base pairs of the DNA. So this nucleosome, the DNA which is surrounding the nucleosome, the total length of the DNA will be 200 base pairs. Okay, 200 base pairs. The chromatin is the repeating unit of nucleosome. Then thread-like stained colored bodies is seen in the nucleus. So nucleosomes in chromatin, they look like beads on the string. So I wanted to look at this picture. This picture I wanted to see where the chromatin network looks like, in the presence of nucleosome, it looks like beads on the string-like structure. Okay. So that's about the packaging of the DNA. Now let us talk about the different regions in the DNA. So when you consider the DNA or the chromosome, you can see like, if you have the chromosome like this, you can see like on staining, some of the parts look very dark and they form bands, and the other parts look very light. Okay. The part which look very dark, they are called as heterochromatic region. What are they called as? Heterochromatic region. And on staining, these heterochromatic regions take up dark stain. Okay, they get colored very darkly compared to the light region, the lightly stained region, which are called as the euchromatic region. What are they called as? Euchromatic region. If we have to look at the chromatin level, you can see that this is how they look. The euchromatic region is loosely coiled and heterochromatic region is tightly coiled, and that is why it is highly condensed here in case of heterochromatic region. And that is the reason that it will take up more of stain and it look dark. It look like dark bands on the chromosome. Whereas the euchromatic region is like loosely packed region and it consists of active genes. Okay? It consists of active genes. Okay? And also you can say that this is transcriptionally active area. So euchromatic region is transcriptionally active. Okay, because transcription can easily occur because it is loosely coiled and unwinding becomes very easy, okay, during the formation of mRNA. Whereas here it is highly coiled, and in these regions it is said that like the genes are not very active. Okay, so they are not very active. So let us see the difference here. They have written euchromatic region transcriptionally active, heterochromatic region transcriptionally inactive. Euchromatic region, loosely packed. Heterochromatic region, densely packed. Like this, they are very high, like we would say they are condensed. Then stain slightly. Why? Because they are loosely packed and the amount of DNA will be less. And stains darkly because they are highly condensed, hence they take up more time. So that's about the structure of the DNA. I hope you all understood. Thank you.